So next speaker, Jonathan Herman, he was supposed to be present, but unfortunately at the last moment, he had to stay home. So he's a physician, doctor in infectious disease, and he's a research fellow at the Galid Alter Labs in the Dragon Institute, so Institute Dragon. So Jonathan, are you with us?
Yes, can you hear me?
Okay, yeah, perfect.
Just 1 second while I figure out the sharing of my screen. Great, okay. Right now my video isn't turning on, so I apologize for that. But good evening, my name is Jonathan Herman. I'm an infectious disease doctor and a research fellow at the Alter Lab at the Reagan Institute. Thank you very much for all the organizers for giving me the opportunity to talk at this meeting. I was very excited to join all you guys down in California, but unfortunately Tuesday morning when I was getting ready to pack my bag to get on the flight, I started to have a little runny nose, a sore throat, and came down with COVID. So I'm glad I still have the opportunity to come here and talk to you guys about this topic, and I'm going to be talking about system serology of malaria-irradiated sporozoite vaccines. And so unlike a lot of conferences I talk at where it's all malaria all the time, I'm glad to be the first malaria talk of the conference, and I can give you guys an overview and a general sense of what we're trying to do in the Alter Lab. So malaria is a global infectious disease that causes significant morbidity and mortality worldwide. Falciparum, Plasmodium falciparum, is the species that infects humans that causes the majority of the morbidity and mortality, and is responsible for hundreds of millions of deaths of children under 5 and pregnant women every year, and millions of cases, especially in sub-Saharan Africa. So this is a significant global health problem that's only been worsened and exacerbated by the COVID-19 pandemic. And so essential for control of malaria is effective drugs to treat malaria, and then also effective vaccines to prevent and also treat symptoms. And so getting to this goal is quite hard because of the complexities of malaria biology. So let me get the laser pointer going. Okay, so I'm going to take you through the malaria life cycle. Mosquitoes that are infected with malaria will bite a human being, and then this liver infecting form, the sporozoite, will then go through blood vessels to reach the liver, reproduce exponentially, and then enter the blood again. This is then the asexual blood stage, where then malaria reproduces again in a constant cycle. And this red blood stage of the form of the disease is what causes the majority of morbidity, mortality symptoms that we associate with malarial disease. And then some of these merozoites will go on to form gametocytes. These are the sexual forms, which then are picked up by mosquitoes and perpetuate the infection worldwide. Now, why do we care about such an important, such a complex life cycle? And the reason is because there's a lot of complexity. It really changes how antibodies that we develop via vaccines or antibodies that we develop as treatments really work, depend on what stage they target. So antibodies that will target the liver stage are going to be antibodies that are helpful for preventing infection. Antibodies and vaccines that target the red blood stage are really going to be for preventing disease and severity. And then antibodies that target the transmission stages are really going to be blocking malaria from perpetuating itself and going into mosquitoes and perpetuating the infection. And so with this framework, people over the past 4 decades have been working on malaria vaccines that either target the blood stage here, the sexual stages here, or the liver stages. Now I'm going to focus in on the liver stage because this is the majority focus of the malaria vaccine development community, because these vaccines have the ability to block infection completely. And the 2 vaccines I'm going to talk about are, one is RTSS and the second is PfSPZ, the sporozoite vaccine that was in my title. So RTSS is the one vaccine that's been WHO approved and is in limited use in sub-Saharan Africa and targets a highly immunodominant epitope CSP that's on the surface of the sporozoite. The sporozoite vaccine, on the other hand, is a live cell vaccine that's been irradiated. And one thing that's quite interesting for the sporozoite vaccine is we have decades of research that have shown cellular immunity, especially T cells, are important, but also antibodies might be important. And on the other hand, in terms of the RTSS vaccine, there's been a lot of evidence to show a lot of the immunity is antibody-based. And so just going back in terms of how we think about vaccines, development of B cells, and then the antibodies that then protect ourselves from it, the dogma of the field has largely been with the vaccine, you direct a B cell to make an antibody. These antibodies are able to target an infectious particle, bind it, and cause neutralization. Ta-da. Unfortunately, the majority of the vaccines that we have in use don't use strict neutralization to protect us. And this includes the COVID vaccines. And so there are neutralizing functions that can predominate in some vaccines, some non-neutralizing functions, and then cellular immunity and other functions of the immune system that seem to be important for protection. And so especially for this audience, I want to focus in on parts of the antibody beyond the Fab that are very important for the ability of the B cell repertoire to protect us. And so a lot of our conference so far, we've talked about the variable domain, the FAB, and it's absolutely essential for antigen recognition, neutralization, opsonization. But when you think about the constant domain, it's not just this inert part that's on the back end of an antibody. It's really directing what an antibody does. Does it lead to phagocytosis, cell cytotoxicity, degranulation or activation of different cells, activate complement, and the list goes on for the rest of the slide. I'm not going to go into all those, but it's quite involved. And the way, at least I think about antibodies, is really the variable domain is the targeting part of the antibody, and the constant domain is kind of like the payload that's delivering the effect of an antibody. And so just putting those two concepts together, when you have, let's say, this bacterial pathogen, these gray bars, come through someone's mucosa and start an infection, they find a target cell in this initial infection, and then the infection can spread and disseminate to the rest of the organism. Now, usually we talk about antibodies as being neutralizers, so we think the antibodies bind to this bacterial pathogen, stop the initial infection, but usually when you have an initial infection, you don't have high titer neutralizing antibodies that are already around. Rather, when we think we have an initial infection, we think we have an antibody repertoire that's already present, recognizes some proteins that are put out on the surface of an infected cell, direct these innate immune cells to target this infected cell, and then help to block spread and further dissemination of infection. And so what does that look like in terms of antibodies directing an innate immune response? You have antibodies target and then bind to proteins that are on the surface of infected cells. And then they really direct different innate immune cells to identify this cell and then respond. And so this can be from monocytes that are getting activated or phagocytosing, let's say bacteria or parts of cells. You can have NK cells that get activated and release cytokines or cause ADCC. And you can have complement deposition. And also you can have activation of antigen presenting cells, which then will help perpetuate and be a bridge to the rest of the adaptive immune system. And so thinking about all the kind of ways that an antibody can direct these functions, a lot of that really comes from different variations that we have in the Fc domain. So first, thinking about the Fc domain that we understand the best, the IgG Fc domains, we have 4 different subclasses. And then we have in the CH2 domain, we have a single end-linked glycan that can have at least 36 different glycoforms that altogether can cause around 150 different functional states that will tune the effector function of each antibody. And so with this framework, we wanted to take this framework of functional antibodies and systems profiling to understand what that means for RTSS, that malaria vaccine. And so the first goal of work that happened in the Alter lab in terms of malaria vaccinology was we wanted to identify the protective mechanisms of a lead malaria vaccine candidate, RTSS. And so I mentioned this quickly, but just to show you, this is a recombinant protein vaccine. And so here, this is the full CS protein, CSP. We take the repeat region and then the C-terminal domain, attach it to Hep B surface antigen, which then forms these virus-like particles that are the basis for the vaccine. And so prior to our work, people knew that antibodies were important for RTSS, but antibody titer alone didn't really correlate that well with protection. And so we took our system serology approach to look for functional antibody profiles that correlated with protection. And we were able to use a Lasso PLSDA model, separate out in light green those individuals in this clinical trial who were protected from those in dark green who were not protected. And we found the features that really defined this separation were the ability of these repeat targeting antibodies in pink to cause monocytes to phagocytose, and then also to cause or for these antibodies to bind to the Fc-receptor 3A. Then looking at one study is interesting, but we wanted to make sure whether these correlates of protection really stood up across multiple studies to know they were kind of unifying and possibly mechanistically relevant. So we looked at two other studies, MAL27 and MAL71, and were able to show that these two functional correlates were able to predict protection in these studies quite accurately. And so this was the initial work in the lab. And so the next goal that we had was we wanted to study a live cell malaria vaccine, this Sporozoite PfSPZ vaccine, so we can identify novel correlates of protection. Now, why do we need to look beyond CSP? Because malaria isn't a virus. It's a eukaryotic organism with 14 chromosomes, 23 megabases of DNA, and over 5,500 protein coding genes. So it's complex, and there's a lot of complexity because it needs to live in the blood, in the liver, and a mosquito, and go all between the three. So there's a lot of specification in terms of what you're trying to target and what's available in the parasite form to intervene in. And so here is a diagram of Sporozoite, the liver infecting form of malaria. And there are different targets that we can target. Micronemes, so this is the subcellular organelle that's responsible for infection and infecting a liver cell. There's a very highly organized surface coat that has different proteins. And then also the parasitophorous vacuole, so that's the membrane that the parasite envelops itself in when it infects a liver cell. So with all this diversity, there are many different targets that possibly could be important. And so to get at this question, we wanted to study a cohort where people were vaccinated with the Sporozoite vaccine, and we could assess what kind of antibody features were associated with protection. And so to do that, we worked on the cohort VRC314. So this is a clinical trial where patients were vaccinated with the Sporozoite vaccine 3, 4, or 5 times, and then they were challenged here at these 4 different points with the malaria to see whether they're protected. Now, as you can see, this is a really great human vaccine trial because we're comparing protection for a vaccine that's given intravenously or intramuscularly, or 3 doses or 4 doses or 5 doses. But it's quite challenging as a human immunology trial because our intervention is changing and we have very distinct time points where we're looking at protection, and not everyone is assayed for protection all the time. And so that's why we wanted to take this kind of large systems approach so we can understand overall correlates of protection. And so I've mentioned system serology. What I want to give you is the sense of how we use this technology in this research. So system serology is a systems immunology approach of looking systematically at the antibody features that are associated with protection or other different categorical phenotypes. And so in this study, we looked at 90 different malaria antigens, predominantly ones in the Sporozoite form. And we looked at antigen-specific class and subclass, ability of antigen-specific antibodies to bind Fc receptors, and then also antigen-specific complement deposition, neutrophil phagocytosis, monocyte phagocytosis, and then NK cell activation. And so altogether, this gives us over 1,600 data points per sample, which means we have a very large high-dimensional data set from which to understand kind of the breadth and depth and in-depth features that might be associated with protection. And so I wanted to bring together all the data together in this heat map to show you some overarching features that we found once we started looking at the data. So just to orient you, this is a heat map where every patient time point is a row and every antibody feature is a column. Here in dark blue, these are all the Fc receptor binding assays that we performed. In orange, this is all the Fc driven functions. And then in green, these are all the isotype and titer antigen specific antibody assays. So we didn't cluster our columns, we clustered our rows. And when we clustered our rows, we saw that the majority of the time points clustered together. And so by seeing this here, this is the sample time point. And here in dark green, we can see most of the dark green time points, the 59-week time points clustered together. And so this was already starting to tell us something about our data, that at least time from vaccination was important. And then when we started looking at the separations between the time points and the different broad strokes of the antibody features, we saw that over here where we have all of our IgM features in the 59-week time points seem to be different than the other time points. And so this was kind of the first hint at what we'll talk about more later about the importance of IgM in this vaccine response. And so next we wanted to take a better look at how the data self-assembled. So we used t-SNE projections to try to see how we can feature reduce into a good visual presentation of the whole antibody profile. And so by just coloring the t-SNE projection by this week of the trial when it was controlled, when it was taken, we could see the 59-week time points clustered together, and then the 0, so the pre-vaccine time point, and then the 22, so the peak immunogenicity time point, co-clustered. Next, when we look at the coloring of the same t-SNE plot just by subject ID, we can see where we're looking at the pre and the peak immunogenicity times here, we can see a lot of the same colors clustering together, showing us that in addition to when the time point is after vaccination, there are a lot of patient-specific features that are quite important. But to get to the heart of the matter of what we really wanted to look into was about protection. And so when we start looking at each of the time points by how long that patient was protected from malaria, we can start to see some patterns. Here in the bottom left, you can see these lime green colored patient time points where people were protected for just 3 weeks. You can see the purple for the people who were protected for 59 weeks, so arranging over here. And then you can see those who are not protected, the 0, are just interspersed completely. So then visualizing that in a different way, just cross-sectionally looking at each of the different malaria challenges, you can see the same patterns of those who are protected just in CHMI1 are in the bottom left. Those who are protected in CHMI2 are kind of more centrally located, and then those who are protected in CHMI3 and 4 are kind of more distantly spread out. And so this really informed us that we thought protection within this clinical trial could possibly be working differently, whether you're early after vaccination, in the middle after vaccination, or late, so up to a whole year after vaccination. And so we decided to take cross-sectional time points to look at what those antibody features were that were associated with protection at each of those times. And so to do this, we used a lasso PLSDA machine learning approach, lasso regression, so we can reduce our features from the 1,600 data points to something more manageable that was most representative of the data, and then our PLSDA, so we can use a supervised method to identify of those lasso selected features which are the most important for protection. And so here's the scores plot of our model. We're able to separate out our protected, these light beige individuals, from those who are not protected, the blue individuals. And this is using protection data from our first protection, so looking early on. And when we looked at the loadings plots, so these are the antigen-specific antibody features that really separate out the two groups and are most likely associated with being protected or not. We can see we have some of the CS protein, that CSP I talked about before, some of those antigen-specific antibody features here. But we also have other proteins, including chitinase, MSP, TSRP, and others. So this is really telling us that early on, protection is complex. Then when we use a similar approach and we're looking at CHMI2, we're able to build a similar model that still separates out those who are protected from unprotected. But when we look at that same loadings plot, we can see all the features that were selected are enriched in the protected, and the majority of them are associated with CSP. And so just because a lot of these peptides are named CSP, but are rather subdomains of it, NAMP6, NAMP3, and then NAMP3 and R32LR, these are all different domains within CSP or parts of the repeat region of CSP that are really driving protection, some by IgM titer, some by C1q, that protein that's involved in the classical complement pathway. And so we were really interested because this wasn't what we expected, because we knew CSP was somewhat involved in that mechanism of protection from this vaccine. And then when we looked at these longitudinal plots, we kind of better understood what was going on. And so just to take you through these, on the x-axis here, we have the weeks of the clinical trial. The y-axis here is the normalized antibody value. And then all the participants in the vaccinees in this trial are then grouped by how long they're protected from malaria. And so here in dark purple, this is the line representing all the patients who are protected from malaria for 59 weeks. And then here in beige is representing all the participants who weren't protected from the get-go. And so as you can see in these plots for complement deposition and then total IgG to NAMP, pre-vaccination, the antibody features are low. At peak immunogenicity, they go up, and we don't see a huge difference in terms of the amount that they go up depending on how long someone's protected. And then they all precipitously drop by 1 year. And so based on this, we thought we had to go out of we need to use a different analysis approach if we wanted to understand what was protecting the folks at 33 weeks or 59 weeks, the long duration protection. And so just to take you through the schematic of what our clinical trial is, we have many participants, and some of them are tested for protection at each of the malaria challenges, but most of them aren't assayed at each of the CHMIs. And so if we're looking cross-sectionally, it's quite challenging. But to take advantage of all this data so we could find those features that are responsible for long duration of protection, we then created this new metric of duration of protection where we're comparing and regressing across people who weren't protected to people who were protected for 59 weeks. And so to take a similar approach, we used lasso and then we used PLS-R where we regress across the duration of protection. We were able to build a model that separated out in light blue the folks who weren't protected from the get-go from the folks in dark blue who were protected for 59 weeks, and you can see quite a nice gradient. We're able to validate this model, and it's performed significantly better than a model where we just permuted the labels. So we think this model has a lot of insight and is relevant to the cohort. And looking underneath the hood at this VIP scores plot showing the antigen-specific antibody features associated with either longer protection in blue or shorter protection in beige, we can see protection is more complex than in the medium time point. We can see some of the CSP features like NAMP3, but there's quite more diversity here. And so the first step we took was the gut check to be, are the antibodies to these malaria targets, are they relevant? Do they make sense for the biology we're dealing with? So we mapped these antigens back to the sporozoite and used publicly available data sets to find the subcellular localization, and then also presence or absence in the sporozoite stage using mass spec databases. And we're able to validate that we have some targets that are located within the micronemes, some targets that are on the surface, specifically CSP, some targets that are expressed and then exported to the parasitophagus or vacuole. And then we have some targets that we know are present based on mass spec data in the sporozoite, but we're not 100% sure where they are. So this is a really good sign. And then going back to the VIP scores plot, we started to look at some of the patterns. And so IgM, the antigen, the antibody that we think about in terms of a short immediate response to infection, seems to be quite important in this longer protection from malaria. And so doing some of the time series analysis and these time series plots, looking at these IgM features like IgM to trap and IgM to GPI anchored micronemal antigen, I'll call this gamma, we can see that the IgMs from peak immunogenicity can increase, and especially in these folks who are protected for 59 weeks, can be higher and then increase over time. And so this was very interesting and surprising, and really got us thinking strongly about the role of IgM in protection from in this vaccine-mediated immunity. Next, looking back at the other hits that came through, we saw TRAP, that protein that we're interested in from the sporozoite, and we saw both the IgM and the IgA1 came up from our approach. And so looking cross-sectionally and just at the peak immunogenicity time point, we can see IgM to TRAP and then C1q binding to TRAP are both significantly enriched in those who are protected. And then antigen antibody complement deposition was trending towards significant, even at peak immunogenicity. Then if we look at the TRAP features over time, we can see both the IgM I showed you before and then in the IgA continue to decrease in quite a large subsection of the vaccinated population from peak immunogenicity on. Then looking back for other signs, we got really interested in the secreted eukinate adhesive protein. So it says eukinate, so that means it's supposed to be a protein that's only in one of the mosquito stages, but we went back and saw that it is expressed in the sporozoite stage. And then when we look at the peak immunogenicity time point, we can see it's significantly enriched in protected individuals for the IgM, and then definitely trending towards increased for IgA, FcAR, and C1q binding. But when you look at the time course data, it's even that much more interesting. So the IgM to the eukinate protein is higher in those who are protected for 59 weeks than others over the whole time course. And then if you look at the Fc alpha receptor binding to this, which is responsible for binding a lot of IgAs, in the folks who are protected for 59 weeks, their Fc receptor binding capacity just does not decrease in a very interesting way. So I know that was a lot of malaria and a lot of biology, but a couple things that I want you guys to take home from this is one, the importance of Fc and Fc-driven features in our understanding of antibody repertoires in human disease states and also in human biology in general. Second is in the sporozoite vaccine and in many vaccine studies, we see that vaccine-mediated protection isn't associated with a whole shift, even if I'm looking at just malaria-specific antibody features, but rather a more specific kind of quality or specific target shift. CSP, this immunodominant protein that a lot of the malaria community has worked on for the past 4 or 5 decades, is associated with early protection in sporozoite vaccinees, but not with long-lasting or later protection. And really interestingly, IgM and IgA to these non-CSP malaria proteins, really this evidence is showing that it's implicated in long-lasting sporozoite-mediated immunity. And so with that, we have a whole bunch of follow-up studies that we're working on, and then other ways that we'd like to broaden this to study different tools and different vaccines within the malaria community, and also apply these types of approaches towards natural immunity so we can inform and figure out how to design the best vaccines to protect everyone worldwide from malaria. And so I want to take a moment to acknowledge first Galit Alter, my supervisor at the Reagan Institute, and then Sanaria, which is the company that's developed the Sporozoite vaccine, and then Sanaria and AID and the VRC with Bob Cedar, who ran the clinical trial, and then our collaborators at MIT and Wellcome Institute and the University of York, and my funding sources, and of course the participants in the VRC 314 clinical trial. And with that, I'll stop and take any questions.
So we are a bit beyond our time. 1 or 2 questions.
Hi, Jonathan. Thanks. That was really interesting and a lot of data. I'm still kind of trying to remember all of it. It went by so quick. But my question has to do with kind of the history of the repeat binding antibodies in the literature of late. And you're probably familiar with how, not all repeat antibodies are equal, and work by Hedda Wardeman's group and Bob Cedar's group have shown that, some repeat antibodies can bind the different tetramers in the repeat region, and some are kind of specific to one kind of tetramer, and that some are protective and some aren't. And Ian Coburn has published on how these different repeat antibodies might even compete with each other in certain cases. And then Hedda Wardeman's made a point that early responses in the anti-repeat repertoire can be different than later responses. So you could have memory competing with a current immune response. And with all of that and the competition that could be going on, or at least that has a strong hypothetical foundation in the literature, is it possible that some of the correlations you're seeing of antibodies to other targets besides CSP, one obvious hypothesis would be that's driving actual function and protection, but is it also possible that those are just competing against anti-CSP responses that might be not protective, and preventing those non-protective anti-CSP responses from competing with memory anti-CSP responses that are protective? It's a very complicated question, but since there's a lot of correlations in there, I'm kind of wondering, and with the competition, whether some things are just preventing bad and non-protective responses from competing with memory good responses. Did that question make sense? Because it was
no, it makes total sense. So the field of anti-repeat responses is fascinating, and there's been a lot of recent evidence, like you summarized very adeptly, about the difference in terms of early and late, and then functional, non-functional, and then the kind of MER, so how many repeats is something responding to, and whether it's coming from the junction or the full or the kind of traditional repeat. But to the crux of your question, which I think is, when you're looking at correlations like this, how much do you know is due to the actual function or antigen antibody specific feature you're measuring, as opposed to, let's say, what it's competing out, or the reverse effect. And so the immediate response I have is you don't know. And so, because with all correlations, you're never 100% sure whether it's causative. And it's always possible, and especially in the field of infectious disease where you're dealing with something like malaria that does a lot of antigenic variation and has a lot of different mechanisms for immune escape, that you are looking at decoy responses that are really non-productive and aren't really protective. And so that's why I'd say looking for patterns is definitely important. And then everything that we find in terms of these correlations has to be validated, on its own. And so some of the things that we do is we do a lot of Fc engineering and monoclonal antibody engineering to validate these hypotheses. We also do a lot of depletions and also single antibody function type of experiments to kind of validate these. And what I'd say is my general sense of the field is CSP is quite important in protection, but the type of antibody response that we form to CSP we seem to form a peak in terms of titer and duration for mechanisms that aren't quite completely understood. And I think that's why a lot of CSP-focused vaccines have had kind of a peak in terms of amount of protection or their duration of protection. And that's why I think focusing on a lot of these non-CSP targets is going to be fruitful.
Thanks, and really good work.
Oh, thank you very much.
Thank you. Thank you very much, Jonathan. We will move to the next talk.